Hello, nurses. So now we're going to talk about genetics and genomics in med-surg nursing. And this is chapter 5 in your book. So why are we learning this in the first place? So it's important for nurses to understand the genetic basis of disease because most of the serious and common adult onset disorders have a genetic component. So if you'll remember, like throughout the semester, in each chapter, there have been different little excerpts about genetic composition of certain diseases that we've talked about. For instance, when we were in the GI chapter, familial polyposis that is found on, like during a colonoscopy, it's just a mass of polyps in someone's colon. Or Crohn's disease has a genetic component. We talked about the BRCA genes when we talked about breasts with female and male. We talked about diabetes. So all of these things have a genetic component. So it is important that nurses understand at least a little bit about genetics. So the Human Genome Project was a research project that was done on an international basis among several countries, and they were trying to map out all the genes of the human body so that they could uncover what kind of genetics influence certain disease processes. So this whole map, or all of the genes in the human body, are called the human genome. So because of this research project that they did back in 2003, they made a lot of different discoveries and found out that 9 out of 10 leading causes of death in the United States have a genetic component, and that including heart disease, diabetes, and cancers. So again, why do we have to learn about this stuff? I mean, it's just one more thing that we have to learn. But if you look on page 72 in your textbook, on table 5-1, there's an entire list of different competencies for medical surgical nursing practice. And that doesn't mean that you only have to know this if you work on a med-surg floor. You don't have to know this if you're an OB nurse, or you don't have to know this if you're a pediatric nurse. Well, that's false, because pediatrics, obstetrics, every realm of nursing has, all of those things have genetic basis. So there are a lot of different competencies that we need to have, but we don't need to know absolutely everything about it. So one of the big things is that we need to know the genetic terminology. Like, what is autosomal dominant characteristic? What is an autosomal recessive characteristic? What's a genome or a genotype? What is a phenotype? What's DNA? What's a gene? What's an allele? So all of these are important for you to understand, like what are they? So you can explain, not in real big detail, because you're not going to be a genetic counselor. I mean, you may later if you want to, but right now you're in nursing. But you just have to have like a basic understanding of what this means. So we're going to go through a few definitions. So first of all, genetics. Genetics and genomics, just so you know, are used interchangeably, but they're different. They're a little bit different. So genetics is concerned with general mechanisms of hereditary and variations of human traits. So how are traits transmitted from one generation to the next? Genetics is a branch of biology that studies inheritance. So it's the processes by which parents transmit their genetic load to their kids. Genomics, on the other hand, which is a more broad yet specific term, focuses on the function of all the human DNA, including genes and non-coding DNA regions. So how is a gene expressed or turned on, either with one person or a family? So again, genomics, how a gene is expressed and what effects it has on cell regulation within a person or a family. Genomic healthcare is the application of known genetic variations to enhance healthcare to individuals and families. So one thing I didn't mention with the Human Genome Project is that what came out of that is exactly this genomic healthcare. What they found was that we can use exact drug therapies to get the best patient responses because of this gene therapy. So they call that precision healthcare and precision medicine. That all came about because of the Human Genome Project back in 2003. So heredity. Each parent contributes 22 chromosomes, which are paired with 22 from the other parent. So those are known as autosomes. And they contain genetic material that determines most bodily characteristics. The 23rd pair of chromosomes are the sex chromosomes that determine gender. And I know you've been through this all in OB already, but this is just kind of a recap. So every human somatic cell has a map of genes known as the genome. So at fertilization, as you remember from OB, each parent contributes one sex chromosome. Females always contribute the X chromosome. The dad always contributes either an X or a Y chromosome. So the gender of the child is determined by the type of male sex cell that penetrates the ovum or the egg. So although X and Y sperm are formed in equal numbers, more male babies than female babies are born in all countries. So the X and Y, X plus Y is a male and X plus X is female, and I'm sure you already know that. Research ongoing involves pre-selection of gender of one's offspring. So this is very controversial, but in the future it will probably be an option as to whether someone or a couple they'll be able to determine what sex of offspring they want. So chromosomes. So genes can be described as storage units of DNA. Each gene is a fragment of DNA that later determine the genetic traits of the individual. So chromosomes are specific segments of DNA that contain the recipe for a specific protein. So there's a difference between a gene and an allele. An allele is an alternate form of a gene, and I'll kind of show you that on a picture here. So just so you know that alleles are variations and not mutations of the gene. Some traits have more than 3 possible alleles, but each person only has 2, one from mom and one from dad. So just what is an allele? So this says some traits have more than 3 possible alleles. So if you think about somebody's color of their eyes, let's say, people have brown eyes, they can have blue eyes, they can have green eyes, they have hazel eyes. I know people who have one eye is brown and one eye is blue, or one eye is brown and one is green. So that is just one example of a gene having more than one possible allele, or we can have several. So again, this is what a chromosome is, and they're unwinding this chromosome to show what a gene is. A gene, again, is a segment and contains the recipe for the individual traits. On the other hand, this is an allele. So an allele is the variation of a type of gene. But just remember that it is not a mutation. So for each single gene, there are 2 alleles. They're alternates or variations of that gene. An allele determines how the genetic characteristics will be expressed in any given individual. For example, when we're talking about alleles, the androgen receptor gene has several alleles. One is associated with hair loss in men, so it's baldness or androgenic alopecia. And I was just mentioning there's one for the color of the eyes, where there's a specific allele for the colors green or blue. So these are just possibilities and variations of how our physical traits will occur. Sex chromosomes, genes for sexual differentiation of a person. We already talked about this. So chromosomally, a normal male has an X and a Y, and chromosomally, a normal female has an X and an X. So in pediatrics, you probably learned that sometimes a female might have an extra X, or part of the X is taken off of the gene. There may be variances in males as well. But you might have talked about Turner syndrome, Klinefelter syndrome in pediatrics. I'm not getting into that here, but if you're interested in what those are, if you did not learn about those, you would probably want to look those up, because you may encounter people that you care for in the future, so you would want to know something about those conditions. So again, just more terminology that you should be competent with. A phenotype is the characteristics that can be observed. So what are the physical traits? How does the patient look? That is the phenotype. What's expressed? I guess that's really a better way to say it. What is going to be expressed? Because you can determine somebody's phenotype by a lab test. A genotype is the actual alleles for a trait. The actual alleles for a trait. Recessive traits are expressed only when the person is homozygous for the alleles. So recessive traits are expressed only when the person is homozygous. That means they have to have a recessive gene from their mother and a recessive gene from their father to make that trait. Recessive traits are expressed only when the person is homozygous. So homozygous meaning the same. Dominant traits are expressed whether the person is homozygous. so having the same gene allele, or heterozygous for the gene allele. So if they have a dominant allele, just one, they're going to have that trait. So heterozygous different, homozygous the same. Mutations are small variations in gene sequences. Changes are passed from one cell generation to another. The most devastating gene mutations are the ones that change the amino acid codes, so a proper protein is not made or does not function at all. So what examples do you know of that where a amino acid code is changed so that a proper protein doesn't function or isn't made properly? Think about that. Do you know any? Again, there's probably some that you learned in pediatrics. So I'll tell you that one is sickle cell anemia. So it alters a single amino acid in the protein produced for the hemoglobin. So sickle cell anemia. Phenylketonuria, also called PKU, is a rare inherited disorder that causes an amino acid called phenylalanine to build up in the body. So again, a rare inherited disorder. But I'm sure you learned about PKU in either OB or pediatrics, because we check newborns for PKU tests. We do PKU tests. PKU is caused by a defect in the gene that helps create the enzyme. Again, an enzyme is a protein, so there's a defect in the gene that helps create that enzyme needed to break down phenylalanine. So that's why phenylalanine builds up in the blood. Excuse me. Your book talks about pedigrees. We're not going to go through this. You might have done pedigrees in health assessment. I don't know if you did or not, but a pedigree, and this is all in your book, but it's just a graph of family history for a specific trait or health problem over several generations. And this one is a 3-generation pedigree showing inheritance. And so this is on page 78 in your book. The actual picture is from page 79. So, and here's another standard pedigree. It's showing you what the symbols mean. So when analyzing a pedigree, you ask different questions. Is there any pattern of inheritance that you can recognize, or does the trait appear to come and go sporadically through generations? Is the trait expressed equally or unequally among male and female family members? Is the trait present in every generation, or does it skip one or more generations? And do only affected people have children affected with the trait? So your book goes through patterns of inheritance, and they talk about, on page 78, they start to talk about autosomal dominant patterns of inheritance. So autosomal dominant single gene traits require that the gene alleles controlling the trait be located on an autosomal chromosome. So an autosome, this isn't a sex-linked characteristic, it's on an autosomal cell. A dominant gene allele is usually expressed even when only one allele of the pair is dominant. And then they go through, what are the criteria for autosomal dominant inheritance. So you will see that again. So the trait appears in every generation with no skipping. The risk for an affected adult to pass the trait to a child is 50% with each pregnancy. Unaffected adults do not have affected children, therefore their risk is essentially 0%. They have other terminology in here, which penetrance is one, and that's how often or how well within a population a gene is expressed when it is present. So how often, how well are those traits or genes expressed when they are present? So this is kind of what I want you to know. It says in your book, for example, the gene for Huntington's disease has an autosomal dominant pattern for transmission. Autosomal dominant. This gene is highly penetrant, sometimes called fully penetrant. This means that if a person had the Huntington disease gene allele, his or her risk for expressing the gene and developing the disease is 99.99%. Therefore, a person who has one Huntington disease allele is at high risk for developing Huntington disease. Then they go along to talk about how some dominant gene alleles have reduced penetrance. So the person with that kind of dominant gene has a lower risk for the gene being expressed. So they talk about the BRCA gene mutation that increases the person's risk for breast cancer, but it is not fully penetrant. So having the gene mutation, having these gene mutations like the BRCA gene, does not absolutely predict that a person will develop that trait, such as breast cancer. Like just because somebody has the BRCA gene does not mean they're going to get breast cancer. But as we've discussed before, many people who have that trait will prophylactically do surgical procedures, surgical mastectomies, to remove their breasts to decrease their chance of getting breast cancer. So that's penetrance. Expressivity is the degree of expression a person has when a dominant gene is present. So I like how your book talks about this. It's very interesting. So expressivity, again, degree of expression a person has when a dominant gene is present. So this is a personal issue, not a population issue. So although somebody has this gene present, it's always expressed, so they always are going to have the trait, but some people have more severe problems than others. So, for instance, in your book they talk about cafe au lait spots. Cafe au lait spots. So somewhere in my genetics, I have this gene mutation for neurofibromatosis, because I have one cafe au lait spot that's very, 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 very light. My daughter has a cafe au lait spot that's very, very, very dark. But other people with the same gene mutations develop hundreds of tumors called neurofibromas that protrude through the skin. So that's called neurofibromatosis. I do not have a picture of that on here, but you should look that up to see what. And you may have seen somebody out in public with this particular disorder. I know there is a woman, she lives somewhere in this area because I've seen her in Walmart several times, but she has like these tumors all over her face and all over her arms and hands. So again, some disorders are caused by dominant alleles for genes. Inheriting just one copy of a dominant allele will cause the disorder. Again, I want to point out Huntington disease. This is the case with Huntington disease. Achondroplastic dwarfism. So the endocrine, when we were talking about the endocrine chapter, we talked about like lack of human growth hormone and how that can affect people and how they can be dwarfs, or their growth is altered. This is a genetic disorder. So polydactyly, I hope you learned about that in pediatrics as well, like people being born with extra fingers or extra toes. So people who, again, are heterozygous, are not healthy carriers, they have the disorder just like homozygous dominant individuals. So they're going to have the disorder, but again, sometimes their disorder is not expressed as much, just not expressed as much as another person or another individual. So if you look at that Punnett square there, if only one parent has a single copy of the dominant allele for a dominant disorder, their children will have a 50% chance of inheriting the disorder and a 50% chance of being entirely normal. So sex-linked traits. The Y chromosome has only a few genes that are not also present on the Y chromosome. The X chromosome has many single genes that are not present on the Y or elsewhere in the human genome. Some of the genes are specific for female sexual development on the X chromosome. But there are also several hundred genes on the X chromosome that code for other functions. And again, if you learned about Turner's syndrome or Klinefelter's disorder, you have learned about some of these, possibly some of these functions. Few disorders have X-linked dominant expression. Few disorders have X-linked dominant expression and are not discussed in your book at all. So on page 80, turn to that page, and you will see this again. So because the number of X chromosomes in males and females isn't the same, obviously, the number of X-linked chromosome genes in the two genders is also unequal. Males have only one X chromosome, and as a result, X-linked recessive genes have dominant expression in males and recessive expression in females. Okay, so I'm going to say that again. X-linked recessive genes have dominant expression in males and recessive expression in females. This difference in expression is because males do not have a second X chromosome to balance the presence of a recessive gene on the first X chromosome. So you can see that here, that males are more susceptible to sex-linked conditions than females. And that is why, because they don't have that other X to balance out those traits. And this is even a better depiction of it, how you can see it. So males have only one X chromosome, and as a result, those X-linked recessive genes have dominant expression in males and recessive expression in females. So when we try to figure out the probability of inheritance, we use these Punnett squares. And someone's told me that you've already done these in OB, so we're not going to do any. I'm just going to show you some that I have on here. So we have a mom and a dad. We have a mom dominant for dark hair, brown eyes, unattached earlobes. She has recessive nearsightedness, straight hair, no dimples. Dad's dominant characteristics, he also has dark hair. He has normal vision. Now look at mom, she's nearsighted. Dad has curly hair, and in him, he is heterozygous, but he has curly hair. Mom has straight hair. See, she has straight hair, so hers is a recessive trait. The curly hair is a dominant trait, but dad has curly hair, but he is heterozygous for that. Dad has brown eyes, which is a dominant characteristic, and he is homozygous dominant. Dad has dimples. Mom has no dimples. See where that is recessive? It's a recessive trait. So dad has dimples because he is heterozygous. He has one dominant and one recessive. And then dad has attached earlobes. And so they're talking about how far these earlobes hang down. So mom has unattached earlobes, which is dominant, but dad has attached earlobes, which is recessive. So let's look at their Punnett squares and what are their chances of having kids with these different traits. So earlobes, there is 100% chance of unattached earlobes because mom was dominant, like she had both of the she was homozygous for the dominant gene. So on here we have one dominant parent with a recessive trait, so a heterozygous parent and a homozygous parent for straight hair. Curly hair is the dominant here. So there's a 50% chance that the couple's child will have straight hair and a 50% chance of curly hair. So I think these are kind of fun to do. They're not necessarily difficult. You just have to know what kind of genes each parent has. So again, here's dimples, a 50% chance for not having dimples and a 50% chance of having dimples. Hair color. Both of these individuals were dark-haired, and apparently their parents were because they, or well, maybe not, but they had both had the dominant trait. and both were homozygous. Eyesight, I think I recall mom was nearsighted, so she had both recessive, homozygous, and dad had normal vision, but he had one recessive characteristic. So there's again a 50-50 chance of their child being normal vision or nearsighted. And then here's eye color, and again both of them dominant for brown eyes. So I'm just going to show you something that's really interesting to me. I have taught this in the past, or this is actually from when I taught genetics, when I taught body structure and function in an LPN program. So some of this is from my older PowerPoint that I did. So I just wanted to show you these families or these children. So these girls are twins, and this was back in 2009 when this couple from the United Kingdom, and just remember that they're from the United Kingdom, they recently gave birth, it says in 2009, to another set of twins, one with dark skin and one with light skin, just like these girls. So that is a 1 in 100 million or more chance. So this is Kian and Remy Hodgson Horder, both parents of mixed race. So both of their parents had a parent who was, I think both of them had white mothers and black fathers. So this couple, this mixed race couple, had twins that obviously are, one is white and one is black. And this is them when they're a little older. And of course they're pretty grown up now. So here again is another set, these little boys. This is another set of twins, and again they're from Britain, Leighton and Caden Richardson. The mother is Nigerian English, and the father is white. So, again, this is another set of twins from the United Kingdom, from Britain. The little girls on the bottom, Alicia and Jasmine Singerl, sorry, can't talk. So these little people were born to a Jamaican English descent and a father of German descent. So again, there's a mixed race and a white person or Caucasian. So I just think this is very interesting. Here's another one. Twins Lucy and Maria Aylmer from Gloucester, United Kingdom. So again, United Kingdom. I don't know what's up with the United Kingdom, but here's some more that are from the United Kingdom. So Marcy and Millie Biggs, and then the one that blows my mind are these twins, Allison and Dean Durant's two twin sets. So they've had two sets of twins, but they are both, like one is dark skinned and one is light skinned. But all of these people are from the United Kingdom. So I just find that very interesting. So what are the benefits and risks of genetic testing? The benefits, I have this in color, so take note of that. One of the benefits is to confirm the diagnosis or identify at-risk individuals, and that is on page 80. Under benefits and risks of genetic testing. Risks. Genetic testing results do not change. So when we tell people that they're at risk for certain disorders, people can have different kinds of reactions to diagnoses. And when they know that these genetic results aren't going to change, they can become just extremely depressed or even suicidal. Or make decisions that, toward better health for themselves, or what they would consider better health for themselves. In other words, just an example would be somebody in a family of breast cancer survivors. Maybe the women in that family have all had genetic testing, and they found they had the BRCA gene. Well, they can become depressed, they can become suicidal, but then again, some people can make that determination, well, I'll either face this and see what happens, or I will have prophylactic mastectomies. So it's just an individual thing. So there are a lot of psychological and social risks, lots of risk for family disruption, and some tests can't necessarily predict the future. We can't predict, if we are going to have some kind of genetic disorder just because our parents or our grandparents had it. We cannot predict that. So there are limitations, basically, is what it's saying. So what's our role as a nurse? We are not genetic counselors. There are people who go into genetics just for that purpose of being genetic counselors. We can help people, help them understand, but we're not genetic counselors. So the best thing we can do as a nurse is to be a patient advocate. It's our professional responsibility and our duty to determine whether the patient understands the consequences of testing. Again, if they find out results, they are not reversible. No matter which professional is involved in genetic counseling, a key feature of counseling is to be non-directive. Non-directive. So we provide information about risks and benefits, but we don't influence the patient's decision to do any tests or to not test. That's not our job. That's not what we do. The patient has to make that determination for themselves. But they ask us a lot of times, well, what would you do? Well, it's not up to me because it's not my body. So it's up to the individual. So then once the patient has made that decision, the counselor or the nurse supports the patient and the patient's decision. So being an advocate and supporting the patient. There are a lot of ethical issues involved, like confidentiality, but you all know about HIPAA. But that is crucial to genetic counseling. The results of genetic tests have to be confidential only to that patient, like only the patient can know. They can't be given to family members. They can't be given to other healthcare providers unless the patient wants them to. They can't be given to insurance companies without the patient's permission. So again, that final determination for genetic testing, that final decision has to remain with the patient. All right, so there's just a couple of questions here. A patient who's preparing to undergo genetic testing is talking with the nurse. Which patient statement requires further nursing intervention? My husband and family are very good support systems. The testing results may help me make appropriate plans for my health. Although I'm a bit anxious, I will be glad to know the results of the test. If the test results come back positive, I'm going to hurt myself. Again, just as I was saying, some people will not handle news well. And just remember that is one risk, and we cannot change, we can't change the results. of a genetic test. So the nurse, the answer here, which I'm sure you know, is D. The nurse should always be aware of a patient indication that he or she could accomplish self-harm. So in the case of a patient undergoing genetic testing, it's important that the patient understand and be prepared for possible psychological, social, and or family risks. So the nurse has to immediately intervene if the patient indicates that he or she is capable of self-harm. Second question, which patient would be considered to have the greatest risk for developing a disease or disorder? 19-year-old woman whose mother was recently diagnosed with ovarian cancer, a 60-year-old woman whose mother and sister both developed breast cancer after age 65, 55-year-old man whose father and 2 brothers all experienced MI after age 60, a 40-year-old woman whose mother died of breast cancer at age 38 years and whose sister developed the disease at age 41 years. So this answer again is D, because breast cancer at an early age can indicate a genetic predisposition. So that person's mother died of breast cancer at age 38, and this woman is now 40, and her sister developed breast cancer at 41 years. So that person in D is at greatest genetic risk for developing the disease. So that is all for this installment, and I thank you very much for your understanding with these recorded lectures. Thank you.